Discussions on how inheritance is determined often focuses on genes, this gene, this mutation, etc. However, there are inherited issues and there are disorders caused by chromosomal abnormalities. And so not only are chromosomes essential to talk about in the functioning of a cell, but also in terms of inheritance. There is chromosomal inheritance, which determines many traits. So obviously, genetic students have studied prior in their academic careers that the gametes are haploid. They have one copy of each chromosome. So a sperm and an ovum each has a chromosome 1, a chromosome 2, a chromosome 3, a chromosome 4. And when they then fuse, and then these two haploid sets of chromosomes, each with 23 chromosomes, are joined into a single nucleus. Now the nucleus is diploid with two sets of 23 chromosomes. Why do we focus on these chromosomes? Why is this so important? Well, if one were to consider, say, this erythroblast, the cells which will produce red blood cells need a number of genes. Red blood cells are very specialized cells with specific proteins. And when we consider the genes needed to function, for example, hemoglobin is made of two alpha globin proteins and two beta globin proteins fused together. Well, the beta globin gene is on chromosome 11, the alpha on chromosome 16. As we'll see, the P and Q, they refer to the short arm in P versus Q, the long arm. And then the 15 and 13 refer to the location on those arms. And so the genes which this red blood cell precursor will use are on different chromosomes. So keeping track of the inheritance of chromosomes, whether any chromosome has a deleted region or a duplicated region, it's important. But the effects would vary depending on which chromosomes were duplicated, which portions of each chromosome might be changed, because once again, the Rh factor is an important gene, but that's located on chromosome 1, while the ABO glycosyl transferase, which determines blood type, on chromosome 9. And then we could see that with other genes as well. And so this red blood cell precursor to become a red blood cell is going to require different genes from different chromosomes. And that would then be true of any cell type. So just one more example, if we were to consider this a cell of the breast, and we might be concerned then about breast cancer. There are prolactin receptors on chromosome 5, estrogen receptors on chromosome chromosome 6. If we're worried about the tumor suppressors, P53 and BRCA, they are located on different chromosomes. And so thus, that would be a different lecture if we were to talk about cancer. If there were extra chromosomes or missing chromosomes, the question would then become, well, which chromosomes are extra, which ones are missing? Because the genes located on each chromosome would then vary. And so this could then be said for any cells. So when we look at that at the chromosomes, we give them numbers and we put them in groups. One of the determinants is if we look for the constricted region known as the centromere, where those microtubules, which are known as kinetochore fibers, attach during cell division to help the chromosomes migrate to the opposite poles correctly. If the centromere is about in the middle of, before I do that, the centromere might not equally divide the chromosome in half. And then if one arm is shorter than the other, the long arm is what's called the P arm, the short arm is called the Q arm. And so if we were then to start giving locations on the chromosome, a gene could be located on 5p12. All right, so 12 is a region on the P arm, the short arm of chromosome 5, or 5q19, etc. If the centromere is about in the center of the chromosome, that chromosome is called a metacentric chromosome. If it's close to the middle, but a little closer to one end, it is known as sub-metacentric. And then as the position of the centromere moves farther from the middle, then we have other categories such as acrocentric. And if the centromere is at the end of the chromosome, it is known as a telocentric chromosome. When we look at human chromosomes in general, except for 21 and 22, the chromosomes are labeled with the largest one first, and then as we get to the smaller chromosomes, then they have numbers like 20, 21, and 22. 21 is actually a little shorter than 22, so that's an exception to the rule. The sex chromosomes, which are considered to be chromosome 23, there are two forms, as is covered elsewhere. The X chromosome is about as large as chromosome 4, so it's pretty big, while the Y chromosome is much smaller. It's one of the smallest chromosomes. And so it's the only chromosome where there are two different forms, although only one form in women. The first three chromosomes are what we refer to as group A, where they are the largest of the chromosomes, being either metacentric or sub-metacentric. The group B chromosomes, chromosome 4 and 5, are also large. They're sub-metacentric, so notice that the centromere is not in the middle of the cell. There are a number of chromosomes in group C, and this would include the X chromosome. These are medium-sized sub-metacentric chromosomes. There are the banding that you see reflects differences in how the DNA is packed and condensed. And when we stain for banding, different stains would produce different banding. Now, as I'll show you in the second video as well, while the centromere is a constricted region of the chromosome, some chromosomes actually form a second constricted region as well. And these chromosomes would then be called satellite chromosomes just for having a constricted region. Chromosomes 13, 14, and 15 belong to chromosome D. They are medium-sized acrocentric chromosomes, but they do have that satellite region because of that secondary constriction that they have. Chromosome group E are short metacentric or sub-metacentric. 16 is metacentric, the other two are sub-metacentric, 17 and 18. Chromosome F are even shorter, short metacentric chromosomes, chromosome 19 and 20. And then we have the group G. These are acrocentric chromosomes. These are the shortest. This would include chromosome Y, but 21 and 22, they are also satellite chromosomes for having a second constriction of the chromosome. Now, satellite chromosomes, once again, the reason for this is in addition to the centromere, they have a second condensation, and there might be a reason for this. So the genes here might, for example, be rRNA genes, and the formation of the nucleolus may involve satellite chromosomes. And so the satellite chromosomes can actually have a role in the structure of the nucleus. One of the things which was noted with inheritance is that you can certainly see that certain traits run in families. This family tends to have this trait or that trait. But some individuals who are not in the same family, not related to each other at all, can share traits based on a chromosomal abnormality. So these are profound. A lot of the things we see running in families are single genes, but there are very large numbers of genes which are located on, a chromosome. And so if there is a change in a chromosome where there's an extra one is missing, or there's a change in the structure, this doesn't affect just one gene, this affects many genes. So this individual has a normal diploid count of chromosomes, where they have 23 kinds of chromosomes. That would be 22 autosomes, and then the 23rd pair being the sex chromosomes. But then they have 2 sets of those having been inherited from one set of 23 from the sperm, one set of 23 from the ovum at fertilization. Okay, so notice that this individual is XX. So this would not only be a normal individual, but they would be female. Now, if there is an extra chromosome, or one is missing, or an extra 2 chromosomes, or 2 are missing. So if there's a change from this diploid count, we call it aneuploidy. Right? So an aneuploidy is there is an abnormal number of chromosomes, but it doesn't affect all 23. It just affects 1 or 2 within the set. And one type of aneuploid is what's called a monosomy, where there is only one copy, in this case of chromosome 3. So a chromosome 3 is absent. So this individual, instead of having 46 chromosomes, has 45, because there's only one copy of chromosome 3. In this case, here's a monosomy where there's only one copy of chromosome 13. Here there's a monosomy where there's only one copy of chromosome 18, etc. And so there can be different monosomies depending on which chromosome is missing. There can also be what's known as a double monosomy, where someone is missing 2 chromosomes which are not homologous. So here's a double monosomy, 1,19. So notice there is a chromosome 1 missing, excuse me, and a chromosome 19 missing. That is a double monosomy. Once again, this could affect any chromosome. So you could have a different double monosomy. So here the double monosomy is 915. There's a missing chromosome 9 and a missing chromosome 15. Now, if 2 chromosomes are missing, but they are the same chromosome, they are homologous chromosomes, this is what is known as a nulosomy. So notice that this individual has no copy of chromosome 4. And so there would be consequences of all of these changes, which I'll be going through presently. So one of the things which one can do with patients is, if one suspects that the issue causing the problem the patient is suffering is due to a chromosomal change, you can get chromosomes which are dividing one way to do it is to get blood cells, and you'd need the white blood cells because they have nuclei while red blood cells don't. You'd get them to where they were dividing, and maybe you'd give them a poison which would stop them in mid-division. That way you'd have the most cells to count. You'd make them slightly hypotonic by adding just a little, like water, a solution which is slightly hypotonic. And then, like if you'd have a microscope slide, you drop the cells here, they would burst. and then you'd stain them and look under the microscope, and then you'd see their chromosomes. And then in the old days, you would take a picture, and then you'd actually cut the picture out and then arrange them. Here's the chromosome 1s, because they're the large metacentric chromosomes. Here's chromosome 2, and then you'd line this up in what's known as a karyotype. And then with the picture of this karyotype, you could then talk about any deviations from the normal diploid count of 46. So what you just saw were aneuploidies, where there's an abnormal chromosome count because there is, say, a chromosome missing or 2 chromosomes missing. You could have an abnormal chromosome count because there might be a few extra chromosomes. These are also aneuploidy because it doesn't affect the entire set. That would be a polyploidy. So aneuploidies can include a trisomy, all right, where there would then be 3 copies of a single chromosome. So notice here that there are 3 copies of chromosome 7. This would be known as a trisomy 7. As before, this could affect any of the chromosomes. So here is a trisomy 20. There are 3 copies of chromosome 20, or trisomy 4 a second ago, or trisomy 21. So notice there are 3 copies of chromosome 21. Now, just as we saw before, if you had 3 copies of chromosome 7 and 3 copies of chromosome 16, that would be a double trisomy 716, indicating that there were 2 different non-homologous chromosomes, which non-homologous sets which had an extra chromosome. So that could affect any chromosomes. That was 7 and 16, but here we see 10 and 20. So there are 3 copies of chromosome 10, 3 copies of chromosome 20. That is a double trisomy 10, 20. Once again, this could affect any chromosome such as 4 and 18. If there are 4 copies of one chromosome, so in a double trisomy, there are 2 extra chromosomes. Here there are 2 extra chromosomes, but they are both of the same type of a chromosome. So there are the usual chromosome 20s, but then there are 2 extra chromosome 20s. That is what is known as a tetrasomy. And once again, this could affect any chromosome. And so here you can see a tetrasomy 21 with too many copies of chromosome 21, or a tetrasomy 22. So for reasons which I'll be discussing, all of these are abnormalities which can affect phenotype and thus be a concern in a patient. Also, as I'll discuss presently, while it could affect an entire person, it could also then affect certainly just cells in a particular region, or be involved in cancer cells. The cells are cancerous because of the extra chromosomes or the chromosomal changes that they've undergone. And so here you see normal diploid counts of 23 chromosomes. You saw both the XX female and then the XY male. So notice that in the XX female, the 23rd set, they are identical, whereas in the XY male, the X is a large chromosome, about as big as chromosome 4, while the Y is small, about as big as chromosomes 21 and 22. And so this will also just give examples, so that there are aneuploidies where you're the problem is just an extra chromosome or 2, or you're missing a chromosome or 2. And then these would include monosomies, double monosomies, nullosomies, trisomies, double trisomies, and tetrasomies. This could result because of chromosomes not being sorted out evenly during the process of meiosis, but this can also occur in mitosis. It could occur in the mitosis before gamete production. So while inherited in the sperm or the ovum, it isn't because of a non-disjunction, that's what it's called, in meiosis, but rather in the mitosis of the stem cells, which preceded the ones which underwent meiotic division. So you can see a double trisomy, etc. Now, one of the, this is also covered in the playlist I have on gender. Very often, if you have an extra chromosome, in almost all cases, that will lead to death. There are a few examples where an affected individual might live a number of years. And one example, a trisomy 21, where an individual could live a long life. Certainly there would be effects whose severity can vary. But for the most part, when an individual is conceived with an extra chromosome, that almost always that results in death prior to birth. And the same is true if one is missing a chromosome. Now, some of the variations of chromosomes, there are most commonly seen in the sex chromosomes. And the reason for that is there is a mechanism for cells to turn off an extra X chromosome. Because women have two big chromosomes, which are X's, men have a big one and a little one, a Y. And so to balance out genes, there is already a mechanism where one of these X chromosomes is inactivated. And so therefore, abnormalities are tolerated a bit better. So here, let's focus on the sex chromosomes, because once again, these are more common chromosomal abnormalities. And here you see a here you see a primary spermatocyte, which has just begun meiosis. Now, what should happen after we go through metaphase 1, and then we go to anaphase 1, telophase 1, is that when the primary spermatocyte splits to form two secondary spermatocytes, then the autosomes are evenly divided, being pulled to opposite poles of the cell, and then that would apply to the sex chromosomes as well. And so then when the secondary spermatocyte then goes through prophase 2, metaphase 2, anaphase 2, telophase 2, and then splits a second time, we then get the spermatids, which grow tails and become the spermatozoa. So notice that the spermatozoa should have a sex chromosome, being haploid. So each one should have either an X or a Y, but they should not have two of any of those. But mistakes happen, and a non-disjunction event can occur if those chromosomes are not sorted evenly, as you just saw. So here, let's imagine we are in the primary spermatocyte. And we start to align the chromosomes with the idea that, one pair is going to go to the left, one pair is going to go to the right as we make secondary spermatocytes. If this non-disjunction occurs, all right, so notice that this was a non-disjunction. These chromosomes should have gone to the opposite pole, but they did not divide equally. This occurred in the first round of meiosis, meiosis 1. And as a result, these this is going to split into two, and now both of them will be missing a sex chromosome, while these, the two daughter cells resulting from this cell, will now have an extra one. And so if meiosis occurs in, if non-disjunction occurs in meiosis 1, all of the gametes from that round of cell division are abnormal. So half have no sex chromosome, half have an extra. Okay, now if this wasn't the sex chromosomes, we could talk about any old chromosome. Half now would be missing a chromosome, and the other one would have, the other half would have an extra chromosome. That's what happens if the non-disjunction occurs in the first round of meiosis. If, however, the first round of meiosis occurs normally, All right, so here the primary spermatocyte is going through prophase 1, metaphase 1. Here we are in anaphase 1, and that's occurring normally. We have telophase 1, and now we have two separate cells. Now this primary spermatocyte, the secondary spermatocytes, they have to divide into spermatozoa, but notice that a mistake was made. While the Y chromosomes split correctly, the X chromosomes did not. There was a non-disjunction of the X chromosomes. Now, this was a non-disjunction in meiosis 2. And because of this, half of the cells are normal. They're supposed to have one sex chromosome, and these two have one sex chromosome. But then in the other half are abnormal. One of the daughter cells has no sex chromosome, and one has two. So non-disjunctions can occur in meiosis 1 or meiosis 2. Now, let's just stick with the sex chromosomes for now. If there is a obviously the normal chromosomal differentiation in humans produces ova, which have an X chromosome, and sperm, half of which have an X and half of which have a Y. If an X-bearing sperm fuses with an ovum, we get an XX individual, which is chromosomally female. If a Y-bearing sperm fuses with an ovum, we have an XY individual, which is chromosomally male. Now, sometimes when we consider the sex cells which fuse, a non-disjunction has produced an abnormal number of chromosomes. Here I'm focusing on the sex chromosomes, but this could apply to chromosome 5 just as easily as it could chromosome 10, etc. So if there are no sex chromosomes in one of the gametes, then the individual might be simply X, what is described as a Turner syndrome female. This could have resulted from either the ovum missing the sex chromosome or the sperm missing the chromosome. So this wouldn't necessarily arise from a failure in the gametogenesis of one gender or the other. And so this individual has a monosomy of the X chromosome. They are a Turner syndrome female. But a non-disjunction could also then potentially produce extra chromosomes. So if, say, the sperm or the ovum had two X's, when they fused, you could now have a female who is XXX, sometimes referred to as a meta female. And there can be greater chromosomal abnormalities which are possible in rarer cases. This can also then affect embryos with a male chromosomal gender, because then if the sperm had an X and a Y and it fuses with an ovum, then this individual would be XXY. If the sperm had a Y chromosome, but the ovum had a non-disjunction so that there were two X's, then once again, this could result in an individual who is XXY, and it is possible then to have individuals who are XYY. Now, I'm focusing in this one on chromosomal abnormalities and inheritance in general. I have a separate playlist on gender, which starts first with chromosomal gender and then somatic gender, where the consequences of the abnormalities that you just saw would be discussed. Okay, so I just went through a number of aneuploidies, and this is where there is an abnormality in a chromosome or two. There's one or two extra, there's one or two missing. Another type of chromosomal abnormality is what is known as polyploidy. So when you look at these this cell and its DNA, there are 3 copies of chromosome 1. There are 3 copies of chromosome 2, chromosome 3, etc. And so the there is a whole extra set. This is what is known as triploidy. Now, triploidy could result a couple of different ways. Once again, there could be non-disjunction, say as gametes are being made, where all of the chromosomes end up on one side of the metaphase plate when, I'm sorry, one side of the cell after a metaphase and anaphase. So this extra set of chromosomes can result from a mistake in chromosome movement, a non-disjunction. But also it could result from what's known as polyspermy. So here you have the chromosomes of an oocyte, and here you have the chromosomes of a sperm. And if this one fertilizes this one, then 23 here and then 23 here will result in 46 chromosomes. But notice that there are lots of sperm all trying to gain entry at the same time. If more than one of these were to donate a set of chromosomes, now you would have 23 chromosomes from the ovum, right? That's what you just saw was the cell division to produce the second polar body, which occurs at fertilization. So here's now the true ovum. It was an oocyte before that division. So here you see the ovum, it has 23 chromosomes, and the 23 chromosomes from the sperm will now give the diploid count of 46. But more sperm could have entered in an event known as polyspermy. And this would have then resulted in polyploidy, not an extra chromosome or two, but a whole extra set of 23 chromosomes. There are, on very rare occasions, triploid infants which are born. And more commonly, there are triploid embryos and tetraploid embryos, which would have 4 copies of each chromosome in humans, which would not make it to birth. So triploidy and tetraploidy, they do occur, but are a cause of miscarriage. Very rarely, triploidy might be the situation in an infant born alive, but unfortunately, it would cause death shortly after birth. But there are other ways to view polyploidy. For example, if we were to say, look at liver cells, hepatocytes, we could see that a number of them have, in addition, while some have 46 chromosomes and are triploid, some would have double that and then be tetraploid. And they would not be cancerous. They would that's something that liver cells can do. A bone marrow cell known as a metamegakaryocyte can have 8, 16, 32, or 64 copies of each chromosome. Liver cells can be octoploid. Now, in addition to having whole extra sets, sometimes they have a whole extra set, but they're missing 1 or 2 to make it even more complicated. Liver cells are odd just to make our lives interesting. Sometimes they have 1 nucleus, sometimes they have 2, 3, or even 4 nuclei. And so these abnormal conditions aren't just something that we could discuss with relation to an infant who was just born and trying to study perhaps the cause of a problem, but in normal liver cells, there are different numbers of chromosomes than one would expect in a typical cell. Once again, a lot of these topics can come up in other lectures as well. And so obviously this could affect speciation. So there are polyploid species where there are 2 types of, say, gray tree frog, or 2 types of this, plant, or 2 types of this fish, etc. And if you ask, why are there, why aren't they the same species? It's because that there was an accident where a polyploid embryo developed. Let's say it's tetraploid, but then it was able to, I guess, fertilize itself and then became a new species, so that you have 2 species that we might actually not be able to distinguish, just visually, but nevertheless under the microscope you would see that these cells vary in the number of chromosomes. One organism has double the chromosomal count as another. but as a result, they form different species because they can't reproduce together. This happens in animals, but is relatively rare. It's much more common in plants. And so polyploidy, once again, that's a separate lecture, but polyploidy is a source of speciation, of forming new species rather quickly. These extra chromosomes, they could come from, say, too many sperm fertilizing an ovum from the same species. That would be what's known as autopolyploidy, or even a different species, because obviously pollen can come from different species and land on the stigma of the female part of the flower. That would be called allopolyploidy if they come from different from different species. Inside our bodies, as I mentioned, there are a couple of cell types that have differing numbers of chromosomes. And in general, there are three different mechanisms for that. What is known as endoreduplication is what you would find in those bone marrow cells, which are going to produce the platelets. Here, DNA is replicated, but mitosis does not begin. All right, so there is an S phase in the cell cycle, and then cell division starts, mitosis. But here, cells would undergo the S phase, but not begin mitosis. And as a result, they would have extra chromosomes. Failed cytokinesis is what happens if the S phase duplicates the DNA. The nucleus then divides to produce two daughter nuclei. So there is mitosis. Mitosis is usually accompanied by cytokinesis, which splits the cytoplasm, but not always. All right. And so in fungi, for example, there's lots of coincidic cells where there's just one huge mass of material that has lots and lots of nuclei, because the nuclei undergo division, but the cells don't split. And then even in human cells, those liver cells, if they have two nuclei, it's because the nucleus divided in mitosis, but the cytoplasm did not divide in cytokinesis. And then we have cells in our adult body, which we see, oh, look, here's one cell with lots of nuclei. How did it get that way? Well, the skeletal muscle cell has hundreds of nuclei because hundreds of embryonic myoblasts fuse to make that skeletal muscle fiber. And so you can have cells smushed together and form a larger syncytial cell, which then could have the genetic material from multiple precursors. There's a cell found in bone called an osteoclast, which dissolves bone, which results from multiple monocytes fusing. So it can have, 6 nuclei, 10 nuclei, et cetera, because 6 separate cells fused. Okay. So before I talk about chromosome structure, what does this do? Well, one of the major things that we have to worry about is gene dosage. So here's an individual, and if they have 2 of these chromosomes, then they are making gene product. So, the DNA gets transcribed, it gets translated into proteins. And the amount that you see there, that's the normal amount that you would get if you were expressing a gene from, say, 2 chromosomes. This much red protein, this much blue protein. If you have only half the normal number of chromosomes, if you have a monosomy, now you're only making half as much of the normal amount of protein. If you have an extra chromosome, a trisomy, now you're making extra protein. What would that do? Well, obviously, it would vary from gene to gene, but because we're talking about a chromosome here, a chromosome has lots of genes. So now there are lots of genes whose product is present at only half the normal level, or more than the normal level. And so this then could have an effect on phenotype, because we're talking about gene dosage. If you recall, conversations about cancer, it's not just a question of which genes you're using, but the level to which you express them. All right, so you want to make the right genes, but making too much of a gene product can be bad, even if the gene, even if the protein is perfectly normal. If you make too little, that could be bad. And so once again, diploid cells have a certain gene dosage that they have resulting from expressing genes on two different chromosomes. A cell with a monosomy is making less than the normal amount of gene product. A cell with a trisomy is making more than the normal amount of gene product. And so, let's just take embryos, for example. There are all of these signals in embryos which are dosage dependent, which are saying, here you are in the embryo, that's why you should make an arm over here and a nervous system over here. Like the level of protein that a cell perceives help it realize, oh, because I'm perceiving this much of a protein, I must be in this part of the body where it's appropriate to make this structure, an arm, let's say. If you start to change the levels of all of these proteins from that chromosome, obviously that makes it very difficult for embryos to develop normally. So gene dosage is a big reason that having extra chromosomes or missing chromosomes can affect phenotype. There are other reasons, and I'll get to those, but I would like to bring, let me, no, sorry, never mind. I was thinking of breaking this into two separate videos. Now, let me, before going into some of the other things that could affect, the other ways that this could affect, let me talk about what's called changes to chromosome structure. So there are a number of things which can cause a chromosome to break. I'm sorry, I thought I put that in a different order. So for example, one of the reasons we try not to get too many X-rays is, X-rays could cause a chromosome to break. Heat can cause chromosomes to break. Viruses can insert themselves into human DNA, but in the process, they can cause chromosomes to break. There are things known as jumping genes, transposable elements, which can move about the genome, which could cause chromosomes to break. Chromosomes can break, and as a result, you can have one fragment which still has the centromere. This is known as the centric fragment, and obviously the kinetochore fibers could attach there and guide it correctly during cell division. This fragment is missing a centromere, so that's the acentric fragment, and that would then just be lost in cell division. And so chromosomes can break, and this can then cause a number of changes in structure. So deletions, which I'll begin with, and then duplications, inversions, and translocations. Why is deletions bad? Well, once again, it affects gene dosage. So if this chromosome underwent a deletion, now it should have more genes here. And so the individual with the deletion chromosome, they are missing some of the protein here, where a normal diploid person would be making a gene product from two different genes, whereas now it's only coming from one. But we also would see what's called pseudo-dominance. A lot of mutations are recessive, and you would not have the mutant phenotype unless you were homozygous for that. But if you no longer have the good copy of the gene, then once you have one mutation, you express it. So even though it's a recessive trait in most individuals, with you it's being expressed with only one copy, so it's having what's called pseudo-dominance. So you can express a recessive allele with deletion chromosomes. And then once again, you can affect gene dosage as I mentioned. Okay, so let me go back to, all right. So a number of things can cause chromosomes to break. This could result in a deletion. Now, that deleted portion could actually attach to a chromosome that already has that portion. And so you could end up actually, in addition to chromosomes which are deletion chromosomes, you could end up with a duplication where a chromosome which already has that portion nevertheless has another portion added to it. You could have, so here you would see the effect of a duplication. You would have now multiple copies of the genes, so you would have extra genetic material. After something breaks, it could then move to a different chromosome. So let's say a piece broke off chromosome 5. It could actually stick on chromosome, say, 19. All right, so the genes are still there, but they're on a different chromosome. That's what's called a translocation. A reciprocal translocation would involve, let's say there were 2 breaks, all right? But after the 2 breaks, both reattached to a chromosome, but they reattached to the wrong chromosomes. So that they, in essence, the green DNA which belongs here now is on this chromosome, and the pink DNA which belongs here is on that chromosome. That's what's called a reciprocal translocation. In an inversion, the same genes are present, but after the break, the broken portion was inserted upside down. So notice that the green area was near the centromere before, and now we see the pink area is near the centromere. So these occur. You can tell that they occur often by looking at the microscope. Let's say that there's a fertility clinic and a couple goes and they say we've had multiple miscarriages and we're having trouble conceiving. What might be the problem? Well, they might order them the study of chromosomes, because if there are deletions or duplications, now you have when the chromosomes line up in meiosis, because one homologue has more DNA than the other, you would see bulges because one area would say have to fold. So here there is an extra, here's the normal region. Here's the deletion chromosome, it's missing a part. So in order for the portions of the chromosome to align, this part has to bulge outward, and that can actually be seen under a microscope. The same would be true if there was a duplication. And so once again, a common enough occurrence would be individuals who are experiencing multiple miscarriages. They might undergo a chromosomal study and see that there were missing DNA or extra DNA. Okay. So let me just kind of go through those 4 changes in chromosome structure and what they might mean. So once again, deletion chromosomes, they do affect phenotype. So just like if you're trying to identify what's causing a problem in your patient, sometimes you would say, oh, you have a mutation in this gene. Sometimes you would say, oh, there is a deletion, say here on, and that you might name it. It's a deletion on chromosome 4q. Once again, we name the arms. The short arm is the P arm. The long arm is the Q arm. And so these would have to be named. So for example, if you're trying to diagnose, the cause of disease in a patient or something, you would say, there's a deletion on chromosome 14q or deletion on 5p. And that would matter because if you remember that genes are located in specific places. And so if you're looking for what effects that might have, now you definitely want to consider the genes which are in those spots. And here's an example of a deletion. There is an important gene which helps to prevent cancerous growth. It's called retinoblastoma. It's active in lots of cells, but when mutated, there is a type of an eye cancer that it can cause in children. One of the reasons for this is a deletion chromosome, so that someone only inherits one retinoblastoma gene. This gene stops cancerous cells from dividing. But if you now only have one copy as opposed to 2, a normal diploid individual, even if a mutation occurs later in life, they started off with 2 good copies, odds are now they still have 1, which would still prevent cancerous growth. But if you had a deletion chromosome, which you started like with only one, then if you were to have a, um, a mutation in in this normal version, then you would now not have any normal retinoblastoma stopping cancerous growth, and so now you would have um, then cancerous growth. And so that's an example of, a deletion chromosomes being relevant, say, in cancer research. Um, Researchers use this a lot. So for example, one of the ways to map chromosomes, I describe it elsewhere, is what's called deletion mapping. Say you have a new gene which say causes pink eyes in fruit flies. Well, where is it on a chromosome? You want to map it, because then you can do lots of things with it once you map it. Well, you then get a number of different stock lines of fruit flies which have deletion chromosomes. So let's say they're missing a something there. So if we're trying to figure out where this gene is located, if you have deletion stocks that might be, say, missing this part of the chromosome, that part of the chromosome, um, if there's a deletion stock where the fruit fly is now missing the part of the chromosome where the gene is on, now not this lineage because it lost this part, but if you were to lose this part, then you would see an example of pseudo-dominance now, where the mutation would be expressed because even though the mutant gene is recessive, there would no longer be a good copy of the gene which would compensate for it in a heterozygote. And so here, because the deletion is actually in the area that the original gene was in, then the mutation, which is normally recessive, shows that pseudo-dominance and would be expressed. So that might be a concern in a patient, but once again, can be used in genetic mapping. So the second type of change in structure would be a duplication, where there is now an extra portion of a chromosome attached. And once again, this would obviously affect gene dosage, because now there would be two, blue gene, sections, two red gene sections. And so there would be abnormal numbers of abnormal amounts of proteins which are produced. And once again, you could identify it. This is a duplication on the long arm of chromosome 10 or the short arm of chromosome 18, etc. And once again, going into this next video, it would then affect gene dosage, and which genes were duplicated would then determine the effects. Are they genes which affect cell division, in which case, this might promote cancer, for example. Now, once again, part of another lecture, but duplications, and I'm sorry, the bar, the slit-eyed in flies is a result of a duplication of the bar gene. Duplications of genes in the long run lead to gene families. And so when we study the human genome, we find that we don't have all of these, distinct genes, but rather genes which are similar to others. They seem to be modified duplicates of each other. Sometimes genes can be duplicated individually, but sometimes whole chromosomes or sections of chromosomes can be duplicated. And so if you ask, why do we have, multiple globin genes? So there's myoglobin and hemoglobin, there's alpha hemoglobin and beta hemoglobin. The reason then is that, over time, genes and sometimes chromosomes and chromosomal regions were duplicated. So once again, part of another lecture, but gene duplication is very important in evolution. And so while, chromosomal duplication might affect one person negatively, very often when we ask, what was the origin of this gene family, it was chromosomal duplications. Translocations are when genes from one portion of one chromosome break and are then attached to a different non-homologous chromosome. It can be what's called a reciprocal translocation if each transmits something to the other. So part of chromosome 5 goes to 20, part of chromosome 20 goes to 5. And there are famous ones because there are parts of specific chromosomes which are more prone to break. They're chromosomal breakpoints. And so there are specific cancers which have names or even chromosomes. This is known as, say, the Philadelphia chromosome. If you have a translocation between chromosomes 9 and 22, or Burkitt's lymphoma between 8 and 14. What are the effects? Once again, it depends on what genes are in the area affected by the translocation. And if they are genes affecting cell division like ABL or MYC, then these translocations could cause cancer, while other translocations would potentially then have different effects. One of the things that translocations can do is increase the difficulty in meiosis and perhaps leading to miscarriages. Because now if you think, if there's, say, reciprocal translocations, when chromosomes try to find their partners in meiosis, here's a problem, because the bottom part of a chromosome would partner with one, chromosome, let's say 5, whereas the top part would partner with another partner of a chromosome. So if you look here at the two chromosomes which have undergone translocation, when they partner in meiosis, who are their partners? Well, the bottom part pairs to the pink chromosome, but the top part pairs to the green chromosome. The reverse would be true here. And so in synapsis, now this could lead to abnormal conformations and separations of chromosomes. And so now you can have abnormal chromosome counts in embryos, which could lead to miscarriages, abnormal gene counts, etc. The last of the 4 changes in chromosome structure is an inversion. And you might think, well, if the same genes are on the same chromosome, well, who cares? I mean, why, if this chromosome is still going to have a green, red, blue, and aqua or purple region, why would we care that it's now in the reverse order? Well, in chromosome with chromosome structure, dealing with euchromatin and heterochromatin, different parts of the chromosome could be on or off. So for example, there are individuals who would say have one blue eye and one brown eye, and there are different ways of getting this, but sometimes it's because of an inversion. Both eyes, say, have genes to make brown color, but on one of the during embryonic development, the cells which would develop into one eye, there was an inversion. So the brown color gene is now on a part of the chromosome which is turned off. So you have the same genes in both eyes, but one of them is in a different spot of the chromosome, and it is now turned off. A major problem of inversions is what happens in meiosis when gametes are made. Because in meiosis, crossing over occurs. Actually, crossing over has to occur, right? If crossing over doesn't occur, then the cell dies and doesn't produce gametes. So crossing over must occur for normal meiosis. But if there is an inversion, all right, if there is a pericentric inversion, and a pericentric inversion would involve the centromere. So notice that here we've switched the genes on either side of the centromere, which would be, the pink, here is above the centromere, here it's below. So the inversion affected the centromere. If these line up and try to start cutting and pasting during crossing over, this would now lead to abnormal chromosomes in the daughters, where some of the chromosomes would be duplication chromosomes. They would have too many genes. Some would then be deletion chromosomes. They would have too few genes. So if the inversion is specifically a pericentric inversion, the chromosomes produced after crossing over are deletion chromosomes and duplication chromosomes, which once again could affect gene dosage. So both of these daughter chromosomes have a centromere, but notice one has too few genes, one has too many genes, which could affect gene dosage and have effects. A more serious case is in what's known as a paracentric inversion. A paracentric inversion does not involve the centromere. So it would just say, notice that this goes aqua centromere pink, aqua centromere pink. So the inversion did not involve the centromere, but it did affect the arm, one of the arms. So notice that, the orange, green, lilac, peach, aqua is reversed here. So that did not involve the centromere. That is known as what is a paracentric inversion. If these chromosomes line up and try to do crossing over, here's a problem. One of the daughter's chromosomes will not have a centromere, so it is no way of being pulled to one of the poles of the chromosomes, poles of the cells which are dividing in telophase. So it would then be lost. So that would be lost. This one has two centromeres, so it could actually get pulled to opposite sides of the cell. And so when there are inversions, an individual might be fine, but when they try to make gametes, they would produce many gametes which would have deletion chromosomes, duplication chromosomes, acentric fragments which get lost, or dicentric chromosomes which get ripped apart. And so this would be a common cause of miscarriage. So someone who says, that, we've been trying to, reproduce and we've had repeated miscarriages, it can be because of inversions, because now the embryo would have these abnormal chromosomes with too many genes, too few genes, etc. And it would vary a little whether it was a pericentric inversion, which involved the centromere, or a paracentric inversion, which did not. One quick note, these inversion chromosomes are sometimes called crossover suppressors. And once again, sometimes, in addition to talking about it with regard to human patients, this could be discussed as, say, in genetics as far as a technique. So if you're mapping chromosomes using curly plum dicheat stubble flies, you might notice that, say, on the second chromosome with curly and plum, you would just have to understand your genetic technique, and you would understand, wow, there's no crossing over. I can always make sure that curly is on one of the chromosome 2s, plum is on the other chromosome 2, and they're never on the same chromosome. Well, how can you be sure of that? Well, because these lines that you got from the stock center, they are inversion chromosomes. And so when crossing over occurs during meiosis, the product chromosomes are so bad, but because they have extra genes, missing genes, etc., that the embryos fail. So if there were to be crossing over on inversion chromosomes, the embryos die. For that reason, crossovers are sometimes called, inversions are sometimes called crossover suppressors. They're really not, though. They don't suppress crossing over. Crossing over still happens. It's just that crossing over is likely to kill the offspring as a result. Okay. The final point is that I've been, focusing on, like, if this happened at conception, then all of an individual's cells would have extra chromosomes. So all of their cells, would have, a trisomy or a monosomy or whatever. But there is another way that this can happen. And let me just, look at the sex chromosomes. Let's say there's a non-disjunction, all right, where a Y chromosome goes the wrong way. Well, if that didn't happen in the sperm or the ovum, which resulted in this embryo, if it happened in the first cell division, the first cell was a normal XY, But when one Y chromosome went the wrong way, now half of the embryo is now XYY, half is now X. So there's a chromosomal difference. Not all of the cells of the embryo are the same. As a result, this individual is what we refer to it as a mosaic. Now here, this affected the first cell division, but it wouldn't have to just affect the first cell division. So here, let's say the first cell division occurred normally, and there was a non-disjunction in the second cell division, in which case now half the cells are normal, but then half the cells, or a quarter of the cells have too many chromosomes, a quarter of the cells have too few chromosomes. Or this could happen later. And so individuals can then be mosaics. There is an abnormal, karyotype, an abnormal chromosome count in some of their cells, but not necessarily all of their cells. And so then you want to know, well, how big is the area of their body that's affected? When in embryonic development did this happen, this non-disjunction? All right. Now, not only this could happen with any chromosome, and therefore you could have a mosaic. But note that if it's happening with the sex chromosomes, the cells which are blue, they are XYY, but they are still male, as would all of the purple cells be male. But some of the cells just have an X chromosome, and they would then be female. All right, so cells which have an X and no Y are female cells. And so this individual's not only a mosaic, where there's a difference in different parts of their body, but there's actually a difference with, the gender associations with the phenotype of different cells. Let me just get back to that in a second. So non-disjunction can occur after conception, all right, which would just affect then certain cells of the body. And so then this individual is a mosaic because the abnormality of the chromosome would just affect these cells in the indicated region, which descended from that first cell where the non-disjunction occurred. So there are a mix of the normal and abnormal cells. But once again, if this affects the sex chromosomes, then it could potentially affect gender. Now, not so if, let's say, development occurs, so this individual is a mosaic known as a gyandromorph, who has one, say, elbow, which is XY and developing as a male elbow does, and one elbow, which is X, developing as a female elbow does. Well, there would be no noticeable change here because male and female elbows don't differ. But if the cells, which are now different, if they help to form testes and ovaries, now you could actually have an ovary and a testis in the same individual. The ovary developed from the X cells, the testis from the XY or XYY cells. You could actually then have an ovotestis, where one single gonad would have both ovarian and testicular cells with different chromosome counts. This has simply been on, when we talk about inheritance, very often we're talking about you inherit this form of this gene, but chromosomes actually also affect inheritance. And there are chromosomal disorders where you have, abnormal numbers of chromosomes, too many or too few, like maybe one or two too many, that's an aneuploidy, or whole extra sets, that's a polyploidy. But then you can also then have changes in chromosome structure, like deletions, duplications, inversions, and translocations. This could affect a whole individual if this was the state in one of the gametes prior to conception. But if it happens in a non-disjunction after conception, it could then affect some cells in an individual, but not others, in what's known as mosaicism.